Welcome to Biochemistry. Today, we are going to explore a fascinating and essential process in our bodies known as amino acid catabolism. Imagine you are tearing down an old Lego castle to build a brand new spaceship. You cannot just throw the old pieces away. You have to take them apart carefully, sort them, and reuse what you can. In our bodies, dietary and cellular proteins are exactly like that Lego castle, and amino acids are the individual building blocks. Let us dive into how our cells manage this incredible dismantling process. Now that we understand the basic premise, let us look at why this process is so critical. When we eat excess protein, or when our cells break down old proteins, we face a unique problem. Unlike fats or carbohydrates, our bodies simply cannot store excess amino acids for later use. Instead, we must dismantle them immediately. This means separating the useful carbon skeleton, which can be used for energy, from the nitrogen-containing amino group, which is potentially very dangerous. This entire dismantling process is highly coordinated. It is a beautiful system designed to extract vital energy while keeping our internal environment completely safe from toxic byproducts. This brings us to the very first step of the cleanup, known as transamination. Think of transamination as a high-stakes game of hot potato, played by the molecules in your liver. The hot potato is the amino group, which contains nitrogen. If this nitrogen is left wandering around freely, it quickly becomes ammonia, which is highly toxic to your brain and nervous system. To prevent this, the body safely passes the amino group from one amino acid to a designated carrier molecule, usually alpha-ketoglutarate. This carrier happily accepts the amino group to become a new molecule called glutamate. By funneling all the dangerous nitrogen into one specific molecule, the body makes the subsequent cleanup process much easier and safer. Now that we know how the nitrogen is funneled, let us look at the mathematical reality of this chemical reaction. The transamination process is governed by an equilibrium constant, denoted as KEQ. Conceptually, this formula tells us the balance between the reactants and the products. KEQ equals the concentration of the products, C and D, divided by the concentration of the reactants, A and B. Here, C and D are the new alpha-keto acid and the new amino acid, while A and B are the original amino acid and the original alpha-keto acid. Because the value of KEQ is typically very close to 1.0, it means the reaction is freely reversible. If the concentration of reactants increases, the reaction pushes forward. If products build up, it easily runs in reverse to synthesize new amino acids. Before we move on to the next step, let us see this transamination process with a concrete example. Imagine a liver cell needs to process 150 molecules of the amino acid alanine. We need to find out how many molecules of the carrier alpha-ketoglutarate are required and how many molecules of glutamate are produced. Because transamination is a strict one-to-one exchange, we use a simple linear relationship. The number of alpha-ketoglutarate molecules equals the number of alanine molecules times 1. Similarly, the number of glutamate molecules produced is also the initial amount of alanine times 1. By substituting 150 into our equations, we find that exactly 150 molecules of alpha-ketoglutarate are consumed and 150 molecules of glutamate are produced. This perfect one-to-one ratio ensures no toxic nitrogen is accidentally released into the cell. So we have successfully passed all the hot potatoes to our carrier, glutamate. This brings us to step 2, oxidative deamination, where it is time to finally defuse them. Oxidative deamination is the process of taking that collected nitrogen and stripping it away from glutamate, releasing it as free ammonia. Imagine a recycling center where all the collected plastic is finally melted down into a raw, usable, but potentially messy liquid. The body does this carefully inside the mitochondria of liver cells, where the toxic ammonia can be immediately contained. This reaction is catalyzed by the enzyme glutamate dehydrogenase. Importantly, as glutamate is oxidized, a molecule of NAD+ is reduced to NADH. This captures high-energy electrons, making this not just a waste disposal step, but a vital energy-yielding one. This energy generation leads us to an important formula to quantify exactly how much fuel we get from this process. Conceptually, this equation calculates the total energy yielded when the NADH produced during deamination enters the electron transport chain. ET represents the total energy yielded in the form of ATP molecules. The variable N stands for the number of moles of NADH produced. The variable YATP represents the ATP yield per molecule of NADH, which is generally accepted as 2.5 ATP per NADH. If the number of glutamate molecules processed increases, the total cellular energy ET increases proportionally. This highlights exactly how protein breakdown fuels the starving cell when energy levels are low. Now that we have our energy formula, let us apply it to a patient in a fasting state. Suppose this patient's liver undergoes oxidative deamination of 40 moles of glutamate to generate energy. We need to calculate the total moles of ATP produced exclusively from this step. First, we recognize the stoichiometry. Each mole of glutamate produces exactly 1 mole of NADH. So 40 moles of glutamate gives us 40 moles of NADH. Next, we substitute 40 for N and 2.5 for YATP into our energy equation. Multiplying 40 by 2.5 gives us a final answer of 100 moles of ATP. Physically, this massive energy boost helps the patient survive the fast by keeping cellular machinery running, proving that waste removal and energy generation go hand in hand. We have now generated free ammonia and extracted energy, which brings us to the final and most crucial step, the urea cycle. Having free ammonia in your cells is like having highly corrosive acid sitting in a bucket inside your house. You cannot just pour it down the drain. You need to neutralize it first. The urea cycle is the liver's specialized chemical neutralization factory. It takes this highly toxic ammonia and combines it with carbon dioxide to create urea. Urea is a safe, stable, and highly water-soluble compound, much like a securely sealed plastic container. Once the ammonia is safely packaged into urea, it can be dumped into the bloodstream, travel to the kidneys, and be safely flushed out of the body. Without this cycle, ammonia would accumulate and cause severe brain swelling. This neutralization process is a matter of life and death, and it comes with a heavy metabolic price tag, which we can calculate using this formula. Conceptually, the urea cycle acts as a metabolic tax for eating protein. CT represents the total energetic cost, measured in high-energy phosphate bonds, which are ATP equivalents. The variable NU is the number of moles of urea synthesized by the liver. The constant 4 represents the biological fact that synthesizing just one molecule of urea requires the cleavage of 4 high-energy phosphate bonds. Therefore, if your protein intake increases, the variable NU goes up, and the energetic burden on the liver, CT, increases dramatically. This is exactly why high-protein diets increase your overall metabolic rate. Let us see exactly how heavy this metabolic toll can be with a real-world example. Imagine a bodybuilder consumes a massive protein shake, resulting in the liver needing to synthesize 15 moles of urea to clear the excess nitrogen. We need to calculate the total cost in high-energy phosphate bonds. We know the synthesis of a single urea molecule costs 4 high-energy phosphate bonds. To find the total cost, we substitute 15 for Nu in our equation. We multiply 15 by the constant 4, which accounts for the ATP used and cleaved during the cycle steps. Multiplying 15 by 4 yields a final cost of 60 high-energy phosphate bonds. This massive energy expenditure explains why digesting high amounts of protein generates significant body heat, as the liver constantly burns fuels to keep the cycle running. Now that we have calculated the costs, let us visualize the entire urea cycle factory in action. On the screen, you are looking at a schematic diagram split into 2 distinct cellular compartments. On the left, we have the mitochondrial matrix, and on the right, the cytosol. Notice how the cycle begins in the matrix, where ammonia and bicarbonate combine, burning 2 ATP to form carbamoyl phosphate. This molecule then merges with ornithine to form citrulline, which crosses the boundary into the cytosol. Follow the arrows in the cytosol. Aspartate enters, burning another ATP equivalent to form argininosuccinate. This intermediate splits to release fumarate and arginine. Finally, arginine is cleaved to release our safe waste product, urea, while regenerating ornithine, which loops back into the mitochondria to keep the cycle turning. We have covered a lot of ground today, so let us summarize this beautifully orchestrated 3-step dance. First, transamination acts as the collector, safely gathering dangerous nitrogen from various amino acids and funneling it into glutamate. Second, oxidative deamination acts as the releaser, stripping that nitrogen away as ammonia in a controlled environment, while simultaneously harvesting vital cellular energy. Finally, the urea cycle acts as the packager, spending energy to convert that highly toxic ammonia into safe, water-soluble urea for disposal. Together, these 3 interconnected processes ensure that our bodies can utilize proteins for energy and building materials without poisoning our brains with toxic waste. You now have a solid understanding of human amino acid catabolism.